Okay. So
It's okay to record it?
yeah, sure. I got it. Okay.
Right. Okay.
So I will start now, right?
Yeah, I will introduce you
please.
first.
Please.
Maybe you can open your camera. Possible.
Oh, sorry.
Yeah. Okay, so Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. So today we have Dr. IG Liu as our presenter. So thank you, IG, for being here in the Zoom meeting. We are
Thank you.
Yeah, we're very happy and very much looking forward to listening to your talk today. So these students, they are from the protein structure and engineering class. So before they have learned about protein structures, and now they're moving towards learning protein engineering techniques, either with genetic mutation or chemical modifications, and maybe a bit of both. So without further ado, I will give the floor to you with your title, Protein Cage Nanoparticles as Drug Delivery Systems, DDS. Yeah, IG, the floor is yours.
Okay, thank you, Rindia. Thanks a lot for your invitation. So, so it's good. Today I will give you introduction about protein cage nanoparticle as drug delivery systems. So actually, the half of the lecture today, I prepared an overview of other material besides our protein cage. So first of all, let me introduce you. I come from Xiamen University. So Xiamen has more than 100 years of history, founded in 1921. So I would like to introduce you. We have 4 campuses. The main campus is picture showing here is called Siming campus. Here's the main building. In front of the main building is Furong Lake. So the main campus is located here on the island. Okay. So on the map, we can see Xiamen is including Jimei, Xiang'an, this island. So the main campus is here on the island. And we also have a Zhuangzhou campus, Malaysia campus, and the Xi'an campus here. Okay, so here's where our department located. So the picture show here is building of our department. Okay, so I would hope to welcome you to visit our campus and our department one day. Okay, now let's start today's topic about protein cage nanoparticle as drug delivery system. Since you all are in the final year, so it's good to give an overview of the drug delivery system based on other material and start from the basic definition of the drug delivery system. So they are technologies that carry drugs into or throughout the body and release a drug in a desired manner and rate. So it enables to selectively deliver drugs to a specific site. So to get the desired tissue, organ, cell, or intracellular. So as we can see that for drug delivery systems, there are many formulas. Among them, oral drug are one of the most common drug formulas that we use, such as tablets and capsules. And there are a lot of natural carriers that we can use for oral drug fabrication. As figures show here, microalgae diatom, like Indonesia tropical marine diatom, maybe you already know that Rindia and her group have been working with. Diatoms are characterized as polar silica, and we can see from the top view and the side view of the diatoms that they have very large surface area on up to 100 square per meter. 100 square per gram. So I was very surprised when I got to know such large surface area that diatom have. So they can load a lot of drug, high loading efficiency, up to 80%. And in this case, we can load a lot of protein or chemical type of drugs. So besides all those micro-sized pores that we can see from the figure, there are actually also nano-sized pores inside of the diatom that we cannot observe from the SEM. So this is scanning electron microscopic technique that we get from the all these figures. And in this way, we can protect the drug from aggregation or degradation to enhance the bioavailability. And the surface of this biosilica can be modified chemically and or genetically. So to have a tunable drug loading and release, as you may well work on in the near future, to display the specific targeting delivery. And there are topical drugs in the form of cream, gel, lotion, and injectable drugs, for example. And the particle, in this case, particles need to disperse very well in the aqueous solution, like COVID-19 vaccine, require high colloidal stability to favor long-term storage and delivery. So for drug delivery, they are mainly divided into passive delivery and active delivery. For targeting delivery of therapeutics, maybe we can organize into 3 different levels, referring primary targeting, secondary targeting, and tertiary targeting. So first of all, drug need to get to the right organ, right? First of all, it needs to get to the right organ to play their role. So in this case, the phytochemical properties of drug would play an important role to affect their biodistribution. Take size as example. So phytochemical properties for drug here would include size, shape, material itself, so would affect the biodistribution. Take size as an example, we can see that different size would result in different destinations. For example, 2.5 to 10 micrometer, the particles would mostly enriched in macrophage. For tumor therapy, it's good to have particles in the size range of 100 to 200 nanometer, because they can result in enhanced penetration and retention effect in tumor, which I will talk a bit later. While particles smaller than 10 nanometer, they would easily accumulate slowly in the bone marrow. So we can see that size would playing an important role here to affect the biodistribution, and the surface property as well is another important factor that we can see. For as we know that the surface, for example, as we know the surface of the cell is negative charge. So in this case, if the particles of the drug is positive charge, they are easier to be bind to the cell. But for the positive charge or nano drug, they are also easier to be cleared in the blood flow. So means that we need to make a balance between the positive and negative. So it's a good way to design a drug is that it's good to have negative charged drug initially. And then once they are passively accumulate in the tumor region, it's good to have a surface charge relation from negative charge to positive charge. So in this case, we can uptake, so the drugs can be uptake by cells easier to enhance the drug uptake and enhance the bioavailability. So after that, drugs need to distinguish after arriving in the tumor region, then drug need to distinguish normal cell from the cancer cell. So as we already know that in this case, surface modification of drug would also play a role. And first of all, we can see from the difference from the normal cell and the cancer cell. We know that cancer cells, they would ignore body's signal to stop dividing. We know that the body has a built-in process that undergo programmed cell death that tells our body to get rid of cells does not need anymore. When our body does not need anymore. But so when our body send out command signals, a normal cell would listen and stop dividing, but cancer cell will not. So they will keep grow and dividing, and because they grow fast, consume a lot of nutrition and oxygen. That's why we can see the shape of the cells are very different from normal cells, which results in poorly aligned endothelial cells that's lacking a smooth surface layer. In this case, particles between, that's also one of the reasons that particles in the size range from 100 to 200 would easily get access into the tumor region and accumulate in the cell, in the tumor region. That's also one of the reasons because of the dysfunctional lymphatic system, means that once particles, they are trapped, once they are entered in the tumor region, they cannot be cleared easily. So this is called EPI effect, the enhanced penetration and retention effect. Okay, so after they uptake by the cell by endocytosis, what is important here is endosome escape. So nanoparticles or nanodrugs now need to escape from endosome to reach the target organelle. However, endosome escape is one of the challenge, right? It's a lot of drugs, or especially for the protein type of drugs, would be easily it's trapped inside of the endosome. But also we know that from the early endosome to late endosome, especially down go to mature lysosome, there's a large pH decrease. From 7.4 is our physiological pH environment, and to earlier endosome 6.5, late 5.5, to lysosome environment 4.5. So under this low pH, especially for those protein type of drug, they can be degraded. Or may be degraded by the enzymes in the lysosome. That's also one of the reasons that it's important to escape from endosome or lysosome. So what I show here is one of the review paper that shows that introduced and summarized different types of endosome escape methods. That if you are interested, I recommend all this literature that you can read and refer to, or maybe come up with a new idea to design or your own system to get the endosome escape. Okay, so here I summarized what we need to consider in each targeting stage. The primary targeting, first of all, getting to the right organ. Secondary targeting, get the right cell type, distinguish normal cell from cancer cell. And tertiary targeting, it's good to target the subcellular to enhance the bioavailability of drugs. So in this case, endosome or lysosome escape is particularly important. So until today, there are many nanodrugs have been developed. And according what I show here is also actually from a review paper, Advanced Drug Delivery Review from 2022. It classified two types of nanodrug system, carrier-based and carrier-free based drug delivery system. So under the nanocarrier-based drug delivery system, it can further divide into four main categories, which is polymer-based nanomedicine, inorganic, like metallic nanoparticle based on drug delivery system, liposome, that's very popular one, and the protein-based nanoparticles. Okay, let's have a brief look on each type of drugs. So since each type are quite popularly used, and depend on which type of drug you are working on, you will choose the specific type, either carrier-based or carrier-free based. So the drug crystals, they are carrier-free based or nano drugs. They are usually formed by hydrophobic nanoparticles, actually. And there are two main strategies to make: top-down strategy by using grading, ball milling. But in this case, especially during the ball milling process, it actually generates high energy, which would damage the property of drugs, leading to high purity of drug. So, and there are also other strategy to make nanoparticle is bottom-up strategy. So this is a very good straightforward way, but of course higher cost compared to the top-down strategy. So also it's more difficult to scale up compared to top-down strategy. But in any case, we have nanoprecipitation method or mini emulsion methods that you can choose. So in this case, because most of the drugs are hydrophobic, so we can't dissolve drugs in the good solvent, good organic solvent. And when we mix with water, we use sonication or stirring. In the most of the case, we use ultrasonication to make nano droplets around the size from 200 to 800, I think. And then we can leave the solvent to evaporate by heating or gas purging. So after the organic solvent evaporate, then in the final stage, we will get nanoparticles with size from 100 nanometer to 200 nanometer, or even down to 10 nanometer. It depends on the parameter that you are parameters that you use for the preparation of these nanoparticles. So parameters means that the drug concentration, the ratio between the water and organic phase, and so on, like sonication, degree, time, and so on. And for the liposomes, this is one of the popular drug that you are familiar with. It's carrier-based or drug. So this one is formed by phospholipid bilayer as hydrophobic part and hydrophilic part form phospholipid bilayer. So that's why in this case, we can load two types of drug, hydrophobic drug at the lipid bilayer and the hydrophilic part at the core of the liposome. So as you know that the liposome, the membrane of the liposome is more or less similar as our cell membrane. That's why they have very high uptake efficiency by the cells. But the problem of this liposome is that they are very high fluidity. So it means that we have drug leaking problem for this liposome type of drug. So to solve this kind of problem, there are other strategy was developed to solve the drug leaking problem is to have polymer polymeric nanoparticle, a nanocarrier. It's called polymer zone. So this also formed by block polymer. It contains hydrophobic part and hydrophilic parts. So in this case, polymers can entangle together and self-assemble into a shell-like structure, similar as lipid bilayer. But in this case, polymers, they would entangle to each other, form much stable nanoparticle compared to liposome. Let's take an example like liposome. For example, the stability of the liposome, they are stable, for example, like for this one. They may be stable in aqueous solution for 1 week. If you would like to store longer, you probably need to store under minus 80 or lower temperature. But polymer zone, they can easily store under room temperature actually. And the problem is, since they have the layer is entangled by polymers, means that they have low permeability for drugs. Once they are loaded in the polymer zone, maybe it's difficult to be released from the polymer zone. So this is one of the problems. So there are also other strategies to solve this kind of problem. Of course, we can always combine the lipid bilayer phospholipid together with this polymeric block polymer to co-assemble into this kind of polymer lipid binary nanoparticles to load drugs. So what I show here is another type of polymeric nanoparticle or nanocarrier. It's called polymeric nanomotor. It's also formed by block polymer. Shows here is PS, PEG. So PS is polystyrene, which is hydrophobic, and the PEG, I guess you all are very familiar with, the hydrophilic. So this can form self-assemble into in the water. Of course, it will hydrophobic part will have high hydrophobic interaction, forms this kind of bilayer and assemble into a droplet. And in this case, it can load platinum nanoparticle. So here is a platinum nanoparticle, work as a catalyst to generate oxygen as a fuel to make the whole nanoparticle move around to enhance the drug delivery efficiency. I don't know, maybe you will be, I'm showing here is because maybe one of you will be interested in to work with this type of nanocarrier, because besides all these polymeric nanomotor, there are also metal-based nanomotor, or you can choose a protein-based or nanomotor that we will introduce later. And the carbon nanotube, actually similar as polymeric nanoparticle, that you can also choose carbon nanotube as nanocarrier to design a nanomotor to deliver drugs. So carbon-based nanomaterials are very popular because they are relatively biocompatible, and they have very large surface area, and carbon is one of the most abundant element on Earth, and they can be easily designed for drug delivery. Okay, and especially for cancer treatment. In this case, so I choose one example. Choose one paper as an example. Show here is multifunctional carbon tube. So what they did here is they stabilize. They first use this Evan blue, which is water soluble molecule, to stabilize this carbon nanotube to make them well dispersed in water, to enhance the long blood circulation time, because carbon nanotube, as we know, if without modification, it's hydrophobic, so they can be easier be cleaned by the blood flow during the blood circulation. So it's good to enhance the hydrophilicity first, modify with Avon blue. By sonication, they can be easily attached on the surface of the carbon nanotube, and then mixed with other protein-based protein. And here is CE6 is another type of fluorescent dye molecule, actually. And this molecule, CE6, it absorbs near IR. That's why what we result here is multifunctional nanoparticle, which can help to enhance the long blood circulation time. And you can follow this bioimaging, this fluorescent signal of this CE6 to follow the destination of the drug in this mouse, in the body. And since this molecule is fluorescent, means that under the laser, it can also give the excited electron to oxygen and convert the oxygen to the oxygen radical, or radical is ROS, that can kill, which will kill the cancers. And of course, the excited molecule, the excited dyes, they will also generate heat. In this case, is the photodynamic therapy and the photothermal therapy combined therapy, that to get the tumor therapy to kill tumor cells. And there are also other type of carriers, for example, quantum dots. So compared to fluorescent dye, quantum dots, they have a lot of advantages. Since so for example, this type of fluorescent dye, CE6, although they absorb the near IR, but the dye molecule, they are not stable, and they are easily photodegraded. Maybe they can survive under the laser for like 2 minutes, but for quantum dots, they have much longer lifetime. So means that photostability is much longer. So you can dynamically follow the destination of the quantum dots in the body as well as in the cells, and as well as in the cells in real time. That's the advantage of using quantum dots. And especially quantum dots, they have very large surface area. Since quantum dots, they usually have size down to 2 to 3 nanometer, up to 10 nanometer. So they have very large surface area to modify, to load the drugs. So by surface modification of functional groups, either imine group or tile group or other type of group, which is tumor environment sensitive ligand, that you can functionalize drug on the surface and then release at the tumor area specifically. And the metal nanoparticles, they are also very widely used. If we take a gold nanoparticle as an example, what I show here is so as an example shown here is ultrasonic triggered drug release process, because as we know, similar to quantum dots, gold nanoparticles also have very large surface area. And with surface modification, we can also load drugs. I take an example here is DADA as a peptide sequence, okay, peptide type of drug that you can bind to the surface of the gold nanoparticle, either by hydrophobic interaction or multiple hydrogen bonding. And in this case, if you load 2 type of drug on the surface of 2 type different size of gold nanoparticle, and then they further link each other together, form a gold cluster. Actually, in this case, was formed by specific hydrogen bonding between drugs. And in this case, we can use ultrasonic as a trigger to disassemble all nanoparticles and release drugs. Okay, so here's gold nanoparticle, and then the metal oxide as well. Metal ion, as we know that it's magnetic nanoparticle, and they also can be used as imaging process to get real-time monitoring of the destination, the location of the nanoparticle in the body, as well as the location of the nanoparticles in the cells. So we can know where our nanoparticles are, where's the location of the drug, and to monitor the drug release process. And there's another property of this type of nanoparticle is enzyme mimic activity. That's we need to know. So this besides all this metal oxide, okay, actually gold nanoparticle also have kind of enzyme mimicking property. So in this case, they can help to detect molecules such as glucose. So they can play as a sensor to monitor the glucose concentration in our blood. Besides all drug nanocarrier, means that this type of carrier can play as multifunctional property. So when it depends on how you design it. Okay. So enzyme making property. So means that when we choose a nanocarrier, it's also need to think about which type of properties we may want to have, because not everyone would want to have this type of enzyme making property, right? You only want an inert carrier. You don't want to have any catalytic property at all. So in this case, polymeric carbon-based nanocarrier would benefit compared to metal or metal oxide nanoparticle. So how to design a nano drug, how to choose a nanocarrier is an important thing that we need to think about. So there are key factors we should consider during the nanomedicine related research to be clinically translatable. First, rational design during the research and development stage is critical. So the delivery system shall be easily fabricated and purified with minimal steps, so to minimize the cost that during the process. And rapid and economic screening methods should be also applied to enable a broad and feasible optimization of various formulation parameters of nanocarriers. Okay, so in this case, in vitro and in vivo studies and their correlation of nanomedicine and the careful design of this clinical trial is also critical to ensure the biosafety, and finally to reach the stage of the production and marketing. So with such large variety of nano drug candidates and having an overview of drug delivery system, now let's come to this protein-based drug delivery. As we show here, also presented in this literature, there are mainly 3 types of protein-based nanoparticles: protamine, gelatin, and protein cage, for example, virus-like particles that nanocarrier that we are going to use, and I will mainly focus on this part later. Protamine is this type of, I'm not sure if you are familiar with this type of protein, but it's kind of, they are very small and they are arginine rich, a nuclear protein. Since they are arginine rich, so means that they are positively charged protein. Therefore, they are quite often used for DNA stabilization in nature. And it's good to design as DNA or RNA delivery system. So example show here is that they can have a very high drug loading efficiency. If you are interested, you can go to this paper as well. And in this case, what they studied here is that with protamine as a nanocarrier, they show very high inhibition of T cell activation in the vitro study. And gelatin nanoparticle as gelatin as nanocarrier is also a very common used type of drug carrier. So gelatin, as you may know, they are a collection of peptide and proteins that produced by partial hydrolysis of collagen, and that extracted from skin or bone or collective tissue animals and so on. So gelatin, they can form electrostatic. So example show here that they can form electrostatic interaction or hydrogen bonding with drug molecules for drug loading. So once arrive to the targeted cells or region, gelatin can be degraded by enzymes and release the drug. Of course, there's a risk in this case because since drugs probably would also be degraded by enzymes. And as I have mentioned before, that I will focus on virus-like particle-based protein cage. So why would we choose protein cage? As figure show here, so what we can, in the nature, we can find protein cage, for example, ferritin from 15 nanometer, and they are encapsulating, and bacteriophage and plant virus, CCMV, chlorotic motor virus that I'm studying on now, and TMV is also one of the popular plant virus. So we can see from the scale bar that from 10 nanometer to 100 to 1 micro, almost 1 micrometer for the size range, that you can find different shape and different size of protein-based nanocarriers. And they are easily to be modified and through chemical or genetic modification. They are very stable under wide pH range. Take ferritin as an example. So ferritin is a universal intracellular protein cage, and they are used to concentrate and store and transport iron by means of confined mineralization. Since free iron would cause photon reaction, which will cause the cell death, as you may know. So this is actually a self-protection process mechanism that our body have to store iron in a various tissue compartments. So this ferritin, it contains 24 subunits, form spherical cages with external diameter of 12 nanometer or 14 nanometer, inner diameter of 8 nanometer. And in this case, they have two types of protein, which called light, smaller, heavy protein is larger. And So let's have a look how the structure arranged or self-assembled together. It has two-fold, three-fold, and four-fold of channels, which allows or has pore size around 0.3 to 0.5 nanometer to allow the ions to go through and trap ions inside. It will be interesting to study how they release iron. In the literature, actually, it demonstrates that in the most of the case, ferritin were degraded to release the iron. But whether we don't know yet whether if there's a more smarter way to release iron, it's actually one of the interesting topics to develop on or to further study on. And for ferritin, they are stable in a very wide pH range, as I show here, from pH 10 to down to 3.4. They are very stable in the solution. So if we further decrease the pH, they would disassemble. And if we increase, they would disassemble, okay, from the high pH to low pH down to pH 2. If we increase the pH again back to pH 7, they would reassemble into the cage-like structure. But this reassemble process is not completely reversible. They have partial denatured protein which cannot form a native like they cannot form native like structure, which their pores still remain on the cage. So this is also one of the way to go to study on the release nanocarrier or drug nanocarrier to study how whether we can trap the drug and release drug through the pH trigger. Means that through the genetic modification of the protein unit, you can turn the pH to control the for the protein cage disassemble in a way that you would like. Probably you would like to have protein cage disassemble under pH 4. In this case, you can have. In this case, we need to have a full sequence of the protein unit and modify it specifically. And one of the interesting part for this protein is that it was reported in ACIS Nano actually. 2018, it was reported that for heavy chain ferritin, it's able to cross blood-brain barrier easily. So if you are interested in this type of nanocarrier, especially for this ferritin, I would suggest you to read this paper. And so in this case, in this case, it was said that the protein can bind to the transferrin receptor here. That's one of the reasons that it can easily cross this blood-brain barrier and then go to the brain tumor cells. The interesting part is that this ferritin, this type of ferritin, they would accumulate into the lysosome or brain tumor cell, but they wouldn't accumulate. They wouldn't accumulate in the lysosome or normal cells. This is one of the question parts that I would have. If you are interested in to study on why this is happening, I refer you to go for further study on this type of study. And it says that it can accumulate, it can easily cross the blood-brain barrier and accumulate in the brain tumor. Okay, so here's the one of the imaging proof to prove that they can by following the fluorescence in intensity, it proves that this type of heavy chain ferritin can accumulate into the tumor area. Okay. So for more detail, I would suggest you to go to this paper, and they also compared all these healthy mice and sick ones. Okay, so now let's look into the virus-like particles. And virus protein cage, actually, you are very familiar with, I believe. So virus-like particles are nanoscale structure made up of assembled viral protein that are virus-like particle. Well, virus I show here, these are whole virus. How to say? Infect our cell and during this process as attachment, entry, uncoating, replication, assemble around all this genetic material and release. Why it is important to know this process is because a lot of nano drugs, the designing of a lot of design of nano drugs are virus particle mimicking nanoparticles. I don't know if you understand this, because virus can easily be trapped into the cells, uptake by cells, and they can successfully release a gene. So it means that if we design a nanoparticle, virus cage nanoparticle, mimicking a virus infection process, then we can successfully deliver our genome genome or drugs that's delivered to wherever you would like to. So one of the fascinating drug is bacteriophage. As we can see here, it's one of the fascinating nanomachine that's we can find in the nature. So as you may know that it has a long tail. Once it's bind to the surface of the cells, it's like a nanomachine that it will self-release of this genetic material into the cell So this is a very fascinating process that until now, I think we human beings still cannot copy to make this type of fancy nanomachine to deliver our nanodrug, to deliver the drugs or the vaccine that we would like to. So it's interesting to study the protein cage engineering, as well as to study this assembling process and disassembly process as well. So what I show here is a TM figure of this type of bacteriophage. The one of the example is that is by genetic modification of this of the protein or the tails. Then you can basically design whatever targeting cells you would like to, besides of E. coli. So means that we can achieve the targeting here. The secondary targeting is to distinguish the cell type by modification the surface of the proteins So what I as you may know that for virus-like particle, until they have very long history, that the first or the vaccine was developed in 1796. Okay, and until today, the popular one is COVID-19 vaccine. Okay, here are other rotavirus you may familiar with. HBV vaccine is also one of the popular vaccine all over the world. And there are also plant virus, shows green color here. It's also one of the protein that I'm working with. And yes, here what I'm showing here, as table showing here is COVID vaccine. Probably you may already know there are many type of vaccine that you can find in the world. Besides the COVID vaccine means that you can also easily use those carrier for other type of vaccine, tumor vaccine, for example, that we are now working on in our lab. And another interesting type of therapy is oncolytic viral therapy. So this type of therapy is that this type of virus, they would prefer to live in cancer cell, replicate itself in the cancer cell, and lead to cell death. And in our lab, we have been working on the plant virus protein cage. And for plant virus, they are spherical, also tube-like structures. For example, in our lab, we are working on copycorotic motovirus, which have T3 structure. So it's an icosahedral structure with 108 protein units, and TMV, as I introduced before, that it's one of the most popular plant virus in the world that people are studying on. So this copycorotic motovirus that will lead to the leaves be yellow color. That's why it's called. So that's how the name comes from, actually. So it contains 108 protein units form self-assemble through the hydrophobic and multi hydrogen interactions into T3 icosahedral structure with diameter of 28 nanometer and inner diameter of 18 nanometer. So it's interesting. The interesting part of this type of protein cage is that that they are stable under low pH, as I show here. On the right side of the figure, they are stable under the low pH, pH 5, low ion exchange. If we increase the pH and increase the ion exchange, it will decrease the electrostatic interaction between the RNA and the protein unit. So in this way, we can disassemble the protein cage, actually. And when we are increasing the protein pH 5 to pH 7.5, we are actually also increasing the electrostatic repulsion between the protein unit. That's also one of the reasons. That's why this type of CCMV disassemble under high pH. So, and then we can remove the RNA by calcium ion, by precipitation, and centrifuge. Then we can remove the precipitation of the RNA. Then we can get pure protein dimer. And the protein dimer, they can reassemble into the virus-like particle if we decrease the pH from 7.5 to pH 5. And of course, under this neutral pH, we can also apply negative charge nanoparticle to direct this protein unit to assemble around all the templates, negatively charged the template into spherical structure or rod-like structure, depending on the particle template that we use. So the question is, what is the biodistribution of the protein cage where we lead to? So where the protein cage leads the drugs to? Is it safe to use protein cage as nano platform? Is one of the questions that we would like to ask, because virus-like particle or any other foreign particle may cause the immune response. So actually, for CCMV and other type of protein cage, there are already papers and researchers studied the biodistribution of the protein cage in 2007, very early. So in this work, they studied copychlorotic motovirus and heat shock protein. As you know, that in our body there are many different types of heat shock protein. That's also one of the reasons that they choose heat shock protein as a marker that to compare with this CCMV. And the result they get is that they find out both CCMV and HSP, they have very broad tissue distribution. So if we look here, that we almost cannot find any CCMV particle in green, but they are mainly accumulate in other tissue, in other organ. It's very broad distribution, and they are safe. So one of the problem here, we can see that after 24 hours, they are all gone. Most of the CCMV particles would be cleared. So how to improve the circulation lifetime of the nanocarrier to enhance the bioavailability is one of the hot topics that we are and a challenge that we need to solve. So for CCMV or any other type of protein cage, they all have a functional group on the surface. Take CCMV as an example. For native CCMV nanoparticle, we have almost 2,000 of carboxylic acid on the surface in my group, and also more than 1,000 of amine group on the surface. Of course, you can also always use genetic modification to insert a functional group that you would like. And then you can go for further chemical modification to modify the protein cage to get longer circulation lifetime, or to have targeting property. So modification of the PEG is one of the popular methods, either for the liposome or protein cage-based nanoparticle. That the PEG modification is often commonly used strategy, is one of the commonly used strategy to help to escape immune system and prolong the circulation lifetime. And the advantage of using protein cage, as shown here, is that there are 3 phases that we can modify: exterior surface, interior surface, and interface. So different phases, we can achieve different goals, of course. Especially for the interior phase, it's good for macromolecule loading or small molecule loading. And interface engineering is good for controlled assembly and disassembly. Especially if we would like to escape, to have an endosome escape or lysosome escape. So if we engineer specific enzyme triggered disassemble property or specific pH triggered disassemble property, then we can release drug efficiently. As shown here, that again, this process we can use chemical cross-linking or chemical this exterior surface modification. Sorry, here either we can use to exterior surface modification is good to enhance the location, enhance the blood circulation lifetime, as well as targeting property. So in this case, we can have chemical cross-linking or genetic fusion to get molecules that you would like. So there are many chemical methods that we can choose from: lysosome, lysine conjugation, cysteine conjugation, and so on. And for chemical modification, we can further use polymer conjugation to further stabilize the nanoparticle. Of course, on one hand, we can further stabilize the protein nanoparticle, help it from degradation. On the other hand, it also can help to decrease immunogenicity. So to further increase the circulation lifetime. And here's another example for surface modification. To enhance the bioavailability, show here. Genetic modification is another type of method that we often use, and it's good to insert certain type of or the proteins or peptides that you would like to. And the engineering of the protein cage, as mentioned before, it's good to have a triggered property, so we can disassemble the protein cage on the time that you would like, at a specific time and specific space that you would like to. As we introduced before, for this type of protein, it has pore size of 5 nanometer. Okay, so this type of protein is stable under pH 7.4, and it can disassemble under pH 5. Means that pH 5 is almost the pH of the late endosome, actually. So means that they have high chance to escape from endosome, or high chance to escape lysosome to release the drug effectively. But the problem of this type of protein cage is that although they have 5 nanometer of pores, but 5 nanometer of pores is actually quite large for small drugs. So if pore size are too large, then there's a high possibility have drug leaking problem. And here's another example for interface engineering. So in this case, this work actually published in Angle Wonder 2021. If you are interested, I also again refer to this literature. You can read, you can follow. So here they engineered a specific loop that as the interface of this encapsulating, and this loop is also pH dependent, because in encapsulating they have much smaller pores, so the pore size is smaller than 1 nanometer. So you wouldn't have a drug leaking problem in this case. So with this loop, you can disassemble all this protein cage under pH 5. If we follow the pH change from early endosome to to lysosome or late endosome, we can know that they can successfully disassemble in lysosome or late endosome. They actually can successfully disassemble in late endosome. Means that you can successfully have endosome escape, and successful endosome escape would result in higher drug bioavailability. In this case, it's one of the important factors that we are aiming at, or one of the important goals that we are aiming, because we need to decrease the drug resistance and other problems that we may face. And the engineering of the protein cage is also another type of study that you can have a controlled drug loading efficiency. In this case, is engineering, genetic engineering of the N type of the protein. Then you can have in this case is a non-covalent, so interaction between coat protein and the cargo material. Here is frozen protein. So through this specific interaction between coat protein and frozen protein cargo, okay, take an example. Then we can load the cargo material as you actually can control the number of cargo material inside of the cage by controlling the number of this complex and the coat protein, Y type of coat protein. Of course, there's a disadvantage of this type of method, because through this method, the loading efficient, drug loading efficiency is actually is not that high, I would say, because if we count this coat protein, there are 108 coat protein, right? If each coat protein bind to 2 type of protein type of drug, means that you can only have maybe 100 of protein type of drug that's to be delivered in one cage. And of course, in this case, it's good to have a small molecule type of drug. Then you can have higher drug loading capacity, higher drug loading capacity. In this case, again, you can modify the inner surface property of the cage or tube, and then to enhance the molecule drug loading. In this case, what they modified is to enhance the hydrophilicity of the inner surface, and then to load this palladium-based drug to enhance the tumor capacity, enhance the tumor therapy. And the interesting part is that it also has specific selectivity, which is charge-dependent loading. As we can see that 2+ platinum has much higher drug loading capacity compared to 1+ platinum. And the interesting part of this design is that the drug release efficiency is also pH dependent, as we can see from the middle figure, that the drug release efficiency is much higher on the low pH than on the neutral pH, almost twice higher than the neutral pH. Means that in this case, we can have much higher drug release property in the tumor region. Okay, so here are all the summary, all the examples on the drug delivery system based on the protein cage that we can modify from the exterior surface, interface, and interior face to get the purpose that you would like, which is cell targeting, control the drug release, and higher drug loading capacity. And to achieve this different stage or for the drug delivery, as I mentioned before, that you can have primary targeting first or to get to the right organ, and then by surface modification, you can distinguish from the normal cell to the cancer cell. And of course, in the later part, by again surface modification of the protein cage, you can also achieve the subcellular targeting and endosome escape to improve this improve the bioavailability of the drugs. Okay, so this is actually all for today's lecture. Thank you for listening.
Yeah, thank you, AG, for an excellent talk about, particularly protein cages, although you also mentioned about drug delivery in general. So in the next maybe 15-20 minutes, we will take questions. We can start from the students if you have any questions. Anybody?
Ayo tanya.
Have fun.
Yeah. AG, let me introduce myself.
Hi.
Hi, AG. I am
Hi.
a senior of India in ITB. Your talk is really interesting. Yeah, I am interested in your talk, actually.
Okay, thank you.
Yeah, but my background is not protein assembly, something like that. My background is protein structure. But AG, can you go to slide 51?
51, okay.
Yes.
Let me have a look. Here.
Yeah, this one. AG, why is it the I just want to ask, because you can detect your result in different kind of tissues,
Yes.
from heart until thyroid, but the best one is in thyroid tissues.
Yeah.
So my
Yeah.
question, is it the result, these results are good or not good? Because this is my thinking only. If we want to deliver drugs, it should be specific, right? To the specific tissues, something like
exactly. You are absolutely right that if we would like to have a specific regime, like tumor regime, of course, it's good to enhance the targeting efficiency. So in this work, actually, what they studied is that the property or for example, CCMV itself has.
Oh, okay.
So we first would like to know which organ it will distribute it. Like this one is native CCMV. So if we would like to have a tumor specific targeting, then we would definitely need surface modification or the specific type of peptide or protein to have targeting effect. Yeah.
Okay, understand.
Yeah.
Understand
Yeah.
right now. Oh, okay. But this one is I mean, after 24 hours, we cannot detect, right? So it means this is really a good result, right?
We cannot see it's a good result actually. We actually would like to have longer circulation half-life.
Okay,
yes.
okay.
So that's also one of the reasons it's good to have surface modification (OPG) to increase the circulation lifetime.
Okay. What
Yeah.
kind of PEG? Any kind of PEG? Polyethylene glycol?
I think it will be the yes, it's what kind of PG, but it will be the length dependent.
Oh,
So the
okay.
final property will be length dependent. Yes.
And then we have to check every kind of PEG, right?
Yeah, exactly. We need to check. Yes.
Okay.
The stability of the protein cage would also depend on the how much of the PG or other type of polymer that you modify. And it also will depend on the polymer length that we use.
Okay, understand. Okay, thank you, AG.
Thank you.
Thank you, Bu Is. Now, any question from the
Students,
audience?
please raise your question.
Or you can also write it in the chat, maybe. Yeah, if not, then maybe I would ask about the charge. Yeah, so you also mentioned about the charge.
Oh yeah.
Yeah, because the size, yeah, that's quite known, but the
Quite known.
charge, yeah, I mean size
Charge also kind of known. We are also quite known, right?
for the charge.
Especially for nanoparticles, like gold nanoparticles. There are many studies have been carried out to study the charge effect.
Right. But so in this case, wait, I would like to, yeah. So is it better to have a positive charges on the surface then?
For positive charge nanoparticle,
Mhm.
they actually have very short lifetime,
Right.
circulation lifetime. That's why usually it's not good to have positive charge initially. So the design of the nanoparticles, it's good to have negative charged nanoparticle initially. And then once they reach the tumor regime, it's good to have a tumor environment triggered charge reversion from negative to positive to enhance the drug uptake by tumor cells.
Okay, yeah, but if you are talking about proteins, then normally we have like many charges on the surface. Or are you
Yes,
talking
that's
about
right.
the total charge or?
We are talking about, so because you are modifying the surface of protein cage anyway, right?
Okay, okay.
So
So you
in
have
this
to
case, we can always modify a ligand which is negative charge, but once they triggered by the tumor environment, maybe it can be cleaved by the pH or enzymes in tumor environment to have positive charge exposed.
and
So
that's
in this way,
yeah.
so this is one of the strategies that to enhance the cell uptake.
Okay. And while we are on this slide, so when we have particles bigger than, say, the slide before, yeah, this one.
Yeah.
Bigger than 2.5 micrometer or maybe around 7 micrometer, that means you cannot use it for delivery because they are being taken up by the white blood cells, or?
Yeah, it's too big to.
Oh, it's too big.
Yeah. Also, because especially for tumor therapy, if
Right.
it's micro-sized, it's very difficult to accumulate. So here, for EPI effect, if particles are too big, they cannot go through. Yeah.
The membrane.
Yes, so it cannot go through this endothelial layer, this membrane. It's very difficult to accumulate into the tumor region.
The cells.
Yes. So that's why it's good to have a particle size with a few hundred nanometer.
So around 200 to
Yeah,
300
200. Yes, 200 to 300.
Yeah, protein cages are way smaller, right? Like 20 nanometer.
Our CCMV protein cage.
Yeah, protein cage.
Yeah. It's like 30 nanometer, 20 to 30 nanometer, right? The ferritin is like a 15 nanometer.
Yeah,
Ferritin
ferritin
cage.
is even
Very
smaller.
small. Yeah.
Right. Yeah, so in this case, I think you are talking about injection-based
Yeah,
drugs.
I'm talking about injection. Yeah. We are not work with oral drug yet, so actually I'm not familiar with those process.
Yeah, I also read somewhere, I think with oral delivery, you need to take care of the absorption from the intestines in the
Yes.
stomach.
That's
Yeah.
okay.
So
Also
that's
need to avoid the degradation. So the design would be much more complicated compared with injectable drugs.
right. Yeah. But have you heard about injection of silica base or silica nanoparticles maybe?
Silica nanoparticle, yes.
For injection?
They are plenty. Yes.
Blood injection?
Yes. Blood injection. If for mesopolar silica, there are plenty of papers. Yeah.
But it has to be nanoparticles.
Yeah, exactly. And they are much more stable if you make into small 100 nanoparticle, right?
Right. But
so for injectable, it's good to have a very stable colloid.
Colloid.
Yeah, it's good to have a stable colloid. So that's one of the reasons that it's good to have particle with size from under 500 nanometer, I would say.
If it's bigger than that, then they will be taken up by white blood cells.
Exactly, and they are not stable for storage. They would easily precipitate.
Yeah, that's right.
Yes.
I have another questions, AG.
Yes.
You are very expert in protein assembly. So we are working with a protein assembly using fold-on, for example, and then the other one is beta-annulose proteins.
Yeah.
So my questions is, for example, if our proteins will make oligomerizations, pH-dependent oligomerizations, how can we separate them? We just have any gel filtration methods in the lab. We don't have any centrifugation in the lab. Can you give us suggestions, AG?
With you would like to separate dimer from
we would like separate trimer from the monomer.
Trimer
from monomer.
Yeah, from the monomer.
But we are using protein separification. I think FPLC, so that's fast protein separation.
Yeah, FPLC.
Yes, we just have one. We just have the equipment in the lab. We don't have centrifugation.
But
analytical ultracentrifugation we don't have.
Yeah, that's enough to separate protein units from.
How about the concentration? Because yesterday, 2 days ago, I helped students to try to using apa namanya Rindia kolomnya
size exclusion, the
Sevarose apa?
Superose 6.
Superose.
Superose 6. Yes.
And we cannot separate. I mean, we just use 2 milligram proteins, and then it seems like it lost.
Oh, so it's all lost. You mean?
Yes, it's lost.
Yeah, that's one of the problems that it absorbs protein.
What do you mean of that one?
I mean the material itself. It absorbs protein. Yes.
Column.
Yeah. So it's one of the problems that we cannot. I think it's very difficult to avoid.
Right? So you have to go with higher concentration. You meant
higher concentration, but 2 milligram per milliliter is quite
Yeah,
high.
it's
Yeah,
enough
quite high.
usually, right?
Yeah. We often use this concentration,
Right.
Yeah,
but
yeah.
it also depends on the volume. But the
The
fact
volume
is
is only 2 milliliter.
2 milliliter.
Milliliter, yes, 2 milliliter
That's
only.
quite a lot.
but with 2 milliliter or 2 milligram per milliliter or sample,
No, 1 milligram per milliliter, and the volume is 2
Mm.
milliliter.
2
So it's
milliliter.
2 milligram total.
2
in
milligram.
total
I think
proteins, yes.
you
In
can
2
still get materials out.
Yeah, no, I cannot. We cannot get the
can.
material out. It seems it traps into the resin.
Yeah, so it's one of the problem of using this type of separation method that we
Oh.
always lose protein.
Okay.
That's why actually if you can use the gradient centrifuge, that would be better.
Yes, that's the better
Yeah.
one. Yes,
Yeah.
that's right. But we don't have that machine.
Ultra centrifugation.
Ultra centrifuge.
Yeah. We don't have IG in Indonesia. So
Yeah.
I suggest to Rindia, you have to go to IG labs to
Yeah,
improve.
we have ultra centrifuge here. So
Yeah, I'm sure you have everything.
You have everything, yeah. I mean, you can detect the size, the potential surface of the proteins easily. We don't have much equipment IG here.
So I hope you can come to Xiamen University.
Yeah.
And we can work together on this
Yes,
protein
definitely.
assembling actually.
Right.
We are also very interested in the assembling dynamic actually. By using single molecular fluorescence imaging technique.
Atomic force microscopy.
Atomic no fluorescence. Yes, fluorescence microscopy, but single molecular.
But single molecule.
Yes, single molecule, very high resolution.
Wow.
Can you do it on a solid surface?
It's good to have to work on solid surface. Actually, they prefer to study on solid surface. It's easier to detect on solid surface compared to solution.
Yeah, so actually we were myself and Dr. Risanawati, we had a student and she was trying to refold protein, a refolding of protein.
Yeah.
So I think maybe we can study that as well using the microscope as long as we have fluorescent label, yeah?
Yeah, exactly. Yes.
Okay.
Yeah. So this also one of the directions that we are working on.
Single molecule. So what is the resolution then? Like which scale? Nanometer scale?
Nanometer, yes, nanometer. 2-3 nanometers.
Wow. That's great. So you have to label the proteins at several sites, or how does that work?
You only label one side of the protein, I think. So depend on the fluorescence intensity or either through this FRET.
Using FRET. Okay.
Yeah.
Yeah, but that's interesting, yeah, because we in Indonesia in the group of especially Dr. Risanawati and colleague, they produce a lot of insoluble proteins.
Yes, that's right.
Insoluble protein.
Yes, that's right. Yeah,
so
we have
regularly
a lot of.
Yeah,
we work with chikungunya proteins and
viruses
and non-structural proteins, and then pre-membrane proteins as well from chikungunya, and then from dengue virus as well. All of them are inclusion body.
Yeah, they're all insoluble, so they had to do refolding using urea and stuff, but we do not really study the mechanism, like how they refold.
Refolding process. Okay, so you would like to study how they refold.
Yeah, preferably in the pores of the biosilica.
In the pores of biosilica. Okay.
Inside the pores. Because we had like super preliminary study, and we saw that when these inclusion bodies were denatured and put into the biosilica, they start to refold from the shifting of the tryptophan.
Oh, okay, okay. So is it like a specific property of the biosilica or any type of cage they can refer?
Yeah, we're not sure, but maybe it's just the pores and the opening and the small surface like chaperone. If you are familiar with chaperone, that's how it works as well.
Yeah,
Chaperone.
I know that we are also actually we are also working on that
Working with
actually.
chaperone. Yeah.
Yes, we are working on that.
Yeah, that's one side that we would
Yeah,
like to explore as well.
Yeah.
Okay, actually we have a lot of common interests.
Yeah, of course.
That's Rindia's expertise.
Yeah, we have the same expertise because we're from the same group.
Yeah, you and Rindia IG. You and Rindia have the same expertise.
Yeah.
Because we were trained together. Actually, we started together and then we finished together as well, right?
Oh, okay, that's good.
Yeah.
I was a bit earlier, but then like
1 month.
several
Yeah,
months.
you are 1 month earlier.
I was 1 month earlier. 1 month earlier in starting my PhD, and maybe also 1 month earlier in graduating my PhD.
Yes.
So it will be very nice if we can have long-term collaborations.
Yes,
since
that's right.
we are very
Yeah,
limited in instruments here, and maybe but we
Yeah,
have
yeah.
quite many students.
Yeah,
we have
yeah.
quite many students if we have
Because
fun.
we are actually working on synthetic material like those type of therapy, theraponium mimicking property, right? But if you have biosilica, so we can actually working on that directly. We don't
Right. Yeah. For us, it's natural, like genetically
material.
genetically expressed proteins or the biosilica that we extracted from microalgae. So in our group, we normally use organisms to produce these kind of materials, not synthetically.
Yeah.
Okay.
Yeah.
That's great. It will be a lot of fun to study the mechanism or the folding process.
Oh yeah, and I think
that's
also high impact articles. If you
Yes, exactly. So it's good to have a single molecule.
Oh yes,
that's right. Yeah,
that's ACS nano, another ACS.
SES
or maybe higher, I guess.
Higher than ACS nano? Okay, that's already the highest for me.
AG from Beijing, how far your place is?
From Beijing to my city is around 2.5 hours. 2
By
hours,
plane?
I would say.
By plane.
By flight.
Quite far.
Yes.
Because it's quite far. From in Jakarta to Beijing is not really far actually. It's quite similar to from Jakarta to Japan.
Yeah, Xiamen is closer. It's on the south. So
Xiamen is closer.
It's close. Yeah, it's closer to Indonesia.
But there isn't any plane from Jakarta to Xiamen, right?
There is.
Yes,
there is.
There is?
Yeah,
that's good. Okay,
Xiamen,
and it's very cheap.
Yeah,
that's good.
It's
okay,
like
please,
6
go there.
6 million rupiah.
That's very cheap.
Yeah.
Okay, Rindia, please prepare yourself to go to a lab. Next year.
Next year, hopefully.
Okay, I should open this. Okay.
Right.
Yeah.
Okay. So maybe if you can also inform about possibilities on continuing study in Xiamen, maybe if some of the students would be interested to study abroad, or
you are asking about me?
Yes.
Yeah. Or Xiamen in general. Would they normally accept foreign students, or?
Yeah. We have quite a lot of foreign students here in our campus, also in the main campus as well. They would like to have international collaboration. Actually, we would prefer.
Sure. So you also
the university encourages international collaboration. Actually.
Right. So you also provide scholarships, or?
At this moment we don't have. I think we need to apply for the national funding.
Okay.
Right.
As well as international national funding for the international collaboration, but the competition is very high.
Right.
Okay.
Okay. Maybe that's it for today. Maybe, yeah. Thank you for all of your Yeah, maybe they're shy because this
Yes.
because it's conducted in English, so sometimes also get
Yes,
shy.
AG.
But thank you so
Okay,
much
thank
for
you.
your talk. It was very inspirational for us, and I think it opens also many ideas for
Yes,
future
that's right.
collaborations
Mm.
Yes, yes,
between
exactly.
ITB and Xiamen University. So we will be in touch. Yeah, maybe we will invite
Yeah.
you also to Indonesia someday.
Thank
Yeah.
you. Yes,
Mm.
thank you.
Yes,
Okay.
yes.
Okay,
Yeah. So thank you
bye.
very
AG.
much. See you next time. Thank you, everyone. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam
Bye.
warahmatullahi wabarakatuh. Thank you, AG.
Thank you.
Bye.